Hi, Mr. Lambden. This is my DNA replica that I made, and I am quite a nerd, so I already had some molecular modeling kits that I previously owned, and so I decided to use them. However, my sister has taken some of them, so I had to resort to Play-Doh that she made a few weeks ago. So the Play-Doh for majority represents my nitrogen bases in this scenario. So the green one, light green one, I have adenine, then I have this dark green one, cytosine, I have guanine, and then I also have thymine here. I have the two red connections between the two, which represents my hydrogen bonds, and technically speaking, I should have 2 hydrogen bonds between these two, and 3 hydrogen bonds between these two, cytosine and guanine, because of the way that the 2 pairs of nitrogen bonds or nitrogen pairs are oriented, they have a different number of hydrogen bonds, but I ran out of the red pieces, so I figured I would verbally explain it. The nitrogen bases are also classified into 2 groups, which are purines and pyrimidines. And so the purines are adenine and guanine, so these two, and then the pyrimidines are cytosine and thymine, so these two. And all the nitrogen bases are then connected back to a deoxyribose sugar, which is right here, and it's not a ribose because that's associated with RNA. And then those sugars are then connected to phosphates, which kind of completes the 3 parts of a nucleotide, which would be the phosphate, the sugar, and then the nitrogen base. Between the phosphate and the sugar, there is a covalent bond, which is represented by the white lines or bars that I have in there. And then the deoxyribose sugar is labeled through the different carbons, so it has 5 carbons, the first carbon being around here, the second carbon being kind of in between these two, the third carbon being here, the 4th carbon being here, and the 5th carbon kind of hanging out in the middle of these two in the form of CH2. So because there is a phosphate on the top, or the 5th carbon is facing the top of this strand, this is the 5 end, making this the 3 end. And then on the other side of the strand, it's the opposite flipped, so this is the 3 end and this is the 5 end. Because both sides are flipped or opposite, it makes both of them anti-parallel, so the way that I numbered the carbons here is similar to the numbering on this, but it's kind of like upside down and opposite. So, and I think that's it. I think I got it all. Let me check. I'm checking really quick. Yeah. Okay. So, and then also you can see that there is a double helix so that I have both of the sides. I miss our class, and I miss our jokes and your socks. So hopefully you can share that with me next time. Besides that, have a good day.